Hello, my name is Jacob Moir. Today I'll be talking about RET resistance. So just a little background, we have RET is known as the gatekeeper protein for a multitude of cascades, as you can see. It is rearranged during transfection, a proto-oncogene that codes for that tyrosine receptor kinase. It's involved in thyroid and non-small cell lung cancers, and as shown, it's the gatekeeper for a multitude of pathways. So there was actually a single mutation that produced I think it was like a -10 kilocalorie versus the wild type, which is a -11 docking score, kilocalorie per mole. So it is actually the double mutations, the 738 and the 806, that were notable with a significantly reduced docking score by up to 2 and 3 times, 2 and 3 folds. Of course, the 738 IC50 was lowered, and by a 29-fold difference, and the 806 was a 21-fold difference. I want to talk about the importance. Oh, no, I'll get back to that. So this is the workflow, right? We had the 3D crystal structure, the high throughput virtual screening, glide docking. We got down from 12 compounds to 5 compounds. Did molecular dynamic simulations, and then from here we'd like to continue experimental evaluation. Yeah, I suggested doing the kinase inhibition assay, GLOMAX, and a couple of Western blots, which I'll show later. So this is a part of the approach too, was that here you see the double mutations, which were courtesy of the bioinformatics student. This is from the zinc compound library, and you can see the pretty good docking scores that she chose. She only chose 4. That's all that I have structures for, which is right here. So again, this is the preliminary work. We said that these are the glide dock with the 3D crystal structure, and we produced several docking scores. So as for the biological activity, I suggested we run the kinase inhibition assay with varying concentrations. We expect some result to be in nanomolar, probably less than wherever it went. Something less than this 238 nanomolar and 174 would be, that'd be great. Definitely closer to the original. I think it was like 2 nanomolar for Selpercatinib. Then I also suggested, if RET signals, its most potent signal is for RAS-RAF and continuing down into JAK-STAT. So I suggested we do a Western blot of ERK, STAT3, STAT5, GAPDH, and that should say AKT. Just to check the other side, the PI3K-B or PI3K cell cascade. Where'd that just go? And then for a third thing, if we really wanted to test more, we could test the cell proliferation with by testing cyclin D1, CDK D1, ICAM, again, GAPDH as a control. So originality, there is no drug right now to combat these resistance in RET. So we were able to use, molecular dynamic simulations to sort of combat this. And then again, we'll be performing unique, extensive biological assays. So this is my research plan. Is structural analysis less than 1 month. The simulations approximately 3 months. We do further simulations on 5 compounds, another 3 months. Biological evaluation 1 to 2 months. SAR from the biological evaluation 1 to 2 months. Prepare a manuscript. That probably take a week or 2. The expected result should be the identification of a couple selective RET resistant inhibitors and 1 to 2 publications. And that's it. I'm just going to leave this picture up here.
Okay, so what are the ability test, we can suggest we can test this drug killing the tumor cell. So what kind of tumor cell ability test?
Non-small cell lung cancer.
And what kind of the assay you're suggesting? Ability test.
Oh, the kinase glow.
Kinase. So kinase is level. Now we want to do the cell level.
You want to do a cell level? Yeah, we could do an MTT. Yeah, we could do an MTT. That would probably be I would say to run it at the normal treatment level, like 72 hours incubated. And then yeah, so MTT, that would, then you could do the whole cell lysis. And then I would check for all these different proteins, because if you've done, if you find a decent IC50, wherever I just did. Yeah, so let's say we find a decent IC50, we want to make sure that it's closing off all these proteins. So you could Western blot for those.
Yeah.
Thank you.